Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing DBT JRF Entrance Exam question paper conducted in the year 2023. If you are new to my channel, please do like, share and subscribe. Coming to the first question. Which of the following gene is not a constituent of tDNA in Agrobacterium tumefaciens? They have asked not a constituent of tDNA region. So the gene shouldn't be present in the tDNA region. That gene they have asked. Octopine synthase, that is present in tDNA. Isopentyl transferase, that is also present in tDNA. Virulence gene G. Vir region genes are found outside of the tDNA region. So the answer here is virulence gene G. Then next one. Which one of these organelle sets are generally equipped with their own genomes? So this question everybody knows. It is nucleus, mitochondria and chloroplast. They have their own genomes. Answer is option 3. Then next one. Which of the following molecules are involved in the formation of ATP by substrate level phosphorylation during glycolysis? So substrate level phosphorylation, it is the process of forming ATP from ADP and phosphorylated intermediate. So the way in glycolysis, the steps like 1,3-bis phosphoglyceric acid giving rise to 3-phosphoglyceric acid, phosphoenol pyruvate giving rise to pyruvate are the steps where substrate level phosphorylation is taking place. So here both these options, that is A and C, are correct. 1,3-bis phosphoglycerate and phosphoenol pyruvate. Then what is next question? What is meant by tropophase? So tropophase, it is the phase at which primary metabolites are produced. That is called the tropophase. So here production of primary metabolites, that is option 3 is correct. Next question. Match the following. Euchromatin. Euchromatin is partially decondensed region of the chromatin with active region genes. That is euchromatin. Heterochromatin is 3, that is D3. So A2D3. That you can see in option 1 and 4. Then we will see for condensin and cohesin. Condensin is help in chromosome condensation to further reduce mitotic chromosome to compact bodies. That is condensin. And cohesin is link sister chromatids together immediately after replication. So the answer here is option 4, that is B2, B4, C1 and D3. Option 1 is not correct. Then next one. Again match the following. RCSB. RCSB is Research Collaboratory for Structural Bioinformatics. So it should be Protein Structure Database, A4. Then OMIM. OMIM is Online Mendelian Inheritance in Man. So it should be for it should be a mutation database also, B3. Then DDBJ is Nucleotide Sequence Database, C1. Flybase is Organism Specific Genome Database. It is for Drosophila melanogaster mainly. So the answer here is A4, B3, C1 and D2. Answer is option 2. Then next one. The term biotechnology was coined by. So it was coined by Carl Ericki. Okay. Answer is option 3. Then next one. What will be the molecular weight of the peptide with the sequence ELTTEK following its backbone cyclization to cyclic ELTTEK? Molecular weights of ELTTK are given here. So what you have to do is, first you have to add all the molecular weights for the peptide ELTTEK. and you will get 809. Then now for the formation of ELTTEK peptide bond, 5 peptide bonds are there. So one minute. 5 peptide bonds are there. 1, 2, 3, 4 and 5. 5 peptide bonds are there. So 5 water molecules are released, because for the formation of one peptide bond, one water molecule will be released. So for formation of 5 peptide bonds, 5 water molecules will be released. But in the question they have mentioned about cyclization. So one more peptide bond is formed, which makes 6 water molecules to be released. So molecular weight of water will be 18. So 6 into 18 will give you 108. Now subtract 108 from 809, you get 701. So the answer is option 3, that is 701. Then next question. Electrons from excited chlorophyll molecules of photosystem 2 are accepted first by. So they are first accepted by the plastoquinone. Answer is option 4. Then next one. Regarding thermal denaturation, the Tm of a protein denotes. So denaturation midpoint of a protein is defined as the temperature, that is Tm, at which folded and unfolded states of the protein are equally found. That is called at, or they are at equilibrium. That is called Tm of the protein. So as per the options given here, we will see midpoint of temperature range over which denaturation occurs. That is correct. Temperature at which protein starts denaturing. No. Temperature at which protein denaturation is completed. No. It is at equilibrium between the folded and the unfolded states. So 3 is not correct. Minimum temperature at which protein does not denature. That is also not correct. So the correct answer is midpoint of temperature range over which denaturation occurs. Then next one. EcoR1 enzyme recognizes the sequence GAATTC, a stretch of linear DNA with 6 GAATTC sites upon digestion with EcoR1 will give rise to. So you have got a linear DNA molecule. Suppose this is the linear DNA molecule. These are the 6 EcoR1 sites. 1, 2, 3, 4, 5 and 6. So upon digestion of with the EcoR1, you will get how many fragments? 1, 2, 3, 4, 5, 6 and 7 fragments. So the answer here is option 2, that is 7. Then lichen is an example of symbiotic relationship between. So it is a symbiotic relationship between algae and fungi. This you have been learning from your plus 2 level onwards, or before that also. I don't know, maybe plus 2 onwards you are learning lichens. Which of the following enzyme is not a part of the DNA replication machinery? So DNA helicase, yes, it is needed for unwinding. Then primase is needed for primer synthesis. DNA polymerase, of course it is needed for polymerization reaction. DNA endonuclease is not needed. So which is not a DNA replication machinery is DNA endonuclease. Then next one. How much is 10 raised to minus 23 liter equal to? So 1 liter is 1000 ml. They have given different units, ml, microliter, nanoliter, picoliter. Now from this we have to see which is the correct one. So 1 liter is 1000 ml. First we will see the ml case. So 10 raised to minus 23 will be 10 raised to minus 20 ml. So they have given answer as 10 raised to minus 17 ml. That is not correct. Now 1 liter to 10 raised to 9 nanoliters. So that will be 10 raised to minus 14 nanoliters. That is the correct answer. They have given 10 raised to minus 14 nanoliters. So that is correct. Now if it is picoliters, it will be 10 raised to minus 11 picoliters. So that is also not correct. So then here is option 3. That is 10 raised to minus 14 nanoliters. Then next one. D-alanine and L-alanine they are enantiomers. Answer is option 2. Codon optimization is essential too. So what is codon bias? First we will come to that. Now the genetic code of different organisms, they are biased towards using only one of the codon that encode the amino acid. Now suppose you have got alanine which is coded by GCA, GCG, GCU and GCC. Now suppose some organism like bacteria, they will have preference for only GCU codon of alanine. But if other codons come of alanine, they will not be recognized. Then they can lead to reduced level of protein production or stalling of the ribosome during protein synthesis. So that is called as codon bias. Now this condition, it is usually seen in heterologous gene expression in bacteria. Heterologous means suppose you have a gene from the plant is being tried to expressed in bacteria, it is called as heterologous gene expression. In plant, the codon for alanine may be all the 4 codons. If you suggest, then plant will accept the alanine codon whichever comes and it will be synthesized. But if in bacteria, instead of I told you GCU, if other codons come, they will not be accepted by the bacteria and it will result in stalling of the ribosome during protein synthesis, or they may be incomplete protein production, etc. So that is seen in usually heterologous condition. Now the method of adjusting codons to match with the host codons is called as codon optimization. To prevent this codon bias, you use what is called as codon optimization. Now why do you use codon optimization? Maximize protein yield in heterologous host. 1 is the answer. Clone genes in heterologous host, that you can do cloning even in a heterologous host and keep it there. You need not have to go for protein expression. But if you want to maximize protein yield in heterologous host, you have to go for codon optimization. Suppress protein toxicity, increase cloning efficiency. They both are also not correct. Then next one. What is the molar concentration of dinitrophenol in solution made by dissolving 184.107 mg DNP in 1 liter water? So we have the formula for molarity is equal to 1000 WB into MB into B. So 1000 into molecular mass is 184.107 mg, that is the weight of the compound. Then you have got the molecular mass in the denominator, it is 184.107 grams into 1000 ml. They have given 1 liter water, so convert it into ml, so it is 1000 ml. That will give you convert milligrams into grams first. So it will be 184.107 into 10 raised to minus 3 grams. Substitute that value, you will get 10 raised to minus 3 moles per liter. Now 1 mole is 10 raised to 6 micromolar. So 10 raised to minus 3 molar will be 10 raised to 3 micromolar or 1000 micromolar. So the answer here is option 1, that is 1000 micromolar. Then next one. The common metabolic precursor of amino acids such as proline and glutamine is, so it is alpha ketoglutarate. Answer is option 4. Then acid rain is caused by nitrogen dioxide. Answer is option 4. Then which of the following methods cannot be used for ab initio determination of the 3-dimensional structure of protein? So conventional mass spectrometry you cannot use for determining the 3-dimensional structure of the protein, but X-ray crystallography, NMR and cryo-electron microscope, they can be used. Then which of the following statement correctly depicts the Beer-Lambert law? Absorbance is directly proportional to path length and concentration. A is equal to absorbance equal to epsilon Cl. If you remember this formula, then you will come to know that absorbance is directly proportional to concentration and path length. Epsilon is the molar coefficient, extinction coefficient, molar extinction coefficient. So the answer here is absorbance is directly proportional to path length and concentration. Then, as per section 4 of Patents Act 1970, which of the following is not patentable in India? So, invention related to atomic energy is not patentable in India. Answer is option 3. What is the product formed upon decarboxylation of oxalic acid with release of one molecule of carbon dioxide? So, oxalic acid upon decarboxylation will give you formic acid. Answer is option 3. Next one. Which of the following dyes cannot be used as a fluorescent probe for microscopy? So, trypan blue cannot be used. It is used for identifying whether you have got a live cell or a dead cell. So, trypan blue cannot be used as a fluorescent probe. They have asked especially fluorescent probe. So, that is not correct. Now, next one. A tumor arising from endodermal tissue. So, endodermal tissue, the tumor will be carcinoma. A tumor arising from mesodermal connective tissue. Now, mesoderm will give rise to muscle cells. So, remember mesodermal connective tissue is option is fourth one, that is sarcoma. So, B4. A tumor arising from lymphoid cells. Lymphoid cells is lymphoma, and cancer cells arising from hematopoietic cells that do not grow as a solid tumor, that is leukemia. Answer is option 2. And the answer here is as per the given statements. Correct answer is A3, B4, C1 and D2, that is option 4. Answer is option 4. Then next one. The common precursor of neurotransmitters dopamine, epinephrine and norepinephrine is. So it is tyrosine. Answer is option 3. Which of the following is a marker enzyme for lysosome? So it is acid phosphatase. Answer is option 1. Which of the following enzyme is not involved in protein folding? GROES, protein disulfide isomerase, peptidyl prolyl cis trans isomerase, peptidoglycan transpeptidase is not involved. Why? Because first of all it is found in bacteria, and the peptide crosslinks in the bacterial cell wall, the crosslinks in bacterial cell wall are formed by transpeptidases. So they are not involved in protein folding. Then next one. Which of the following plant organs is not a potential explant for tissue culture? So it is the flower which is not a potential explant for tissue culture. Then next one. How much albumin? They have given the molecular weight must be taken to couple it with 1.8 mg aspirin in a 1:1 mole per mole reaction between the two. So we know that moles N is given by W by M, where W is weight of the compound and M is the molecular weight. So they have asked how much albumin must be taken. So we will give it as X divided by molecular weight of albumin is equal to weight of aspirin, that is 1.8 mg, divided by molecular weight of aspirin is 180. So calculate for X, cross multiply, you will get it as 670 mg. So the answer here is option 2. Then next one. In affinity chromatography, the protein bound to the column can be eluted using a buffer containing. So the target protein in affinity chromatography, they can be eluted by bringing changes in the pH or salt concentration, or also by competition with a free ligand. So here they have given in options free ligand is being given. So the answer is option 4, that is free ligand. Then next one. Rehan made a single stranded DNA with the following sequence 5' GGATCT3 3'. With which of the following single strands will it form the weakest duplex? So first we will see the case of option A. 3' CCTA GA. So that is complementary to this particular sequence what Rehan has used. So that will form a strong duplex. Then next one, option B, option 2. That is 5' AGATCC 3'. That is just the reverse of this particular sequence. See, from here 5' to 3' it is AGATCC to 3'. So that will also form a duplex because it is complementary to the given sequence. Now third one, if you see AGAATT 3'. So this is the sequence that you have, and this is the AGAATT. It is nowhere complementary to this particular sequence. So it will not form complementary base pairing in there, and it will form the weakest duplex. But if you see option 4, they only in one region, that is instead of T they have mentioned it as A, but only one single nucleotide is not forming complementary base pair, whereas all the other nucleotides are forming. But in this option 3, maximum number of nucleotides are not forming complementary base pairing. So it will be the weakest duplex. So the answer here is option 3. Then next one. Indian environmentalist with environmental impact awareness. So Sundarlal Bahuguna. Tehri Bachao Andolan. A1. Sri Rajendra Singh. B. Water conservation. Salim Ali is for conservation of Indian birds, and Mrs. Menaka Gandhi is for animal welfare. This was a very easy match the following. So the answer here is option 1, A1, B3, C2, D4. Then next one. Which of the following amino acid has 2 chiral centers? So out of this answer options, isoleucine is having 2 chiral centers. So this one. This is the structure of isoleucine. Carbon 2. This carbon and this carbon is having chiral centers. This carbon is attached to 4 different other groups. So NH2 here, COH here, then CH3, CH2, CH3 is been attached to this carbon atom. So it is one chiral carbon, and this one, the third one, C3. So it is also attached to 4 different groups. H, CH3, CH2, CH3 and CH, COO, NH2. So carbon 3 is also a chiral carbon. So amino acid with 2 chiral centers is isoleucine. Then next one. The tenure of a registered trademark in India is generally. So it is generally 10 years. Answer is option 3, 10 years. Then next one. Which one of the following, this is section B. Which one of the following is not a type of intellectual property right? So, patent. There are 7 types of intellectual property rights in India. So that is copyright, trademark, patent, geographical indication, plant varieties, industrial design, and semiconductor integrated circuit layout design. So patent is there, articraft is not there. So the answer is articraft, industrial design and geographical indicator. They are also intellectual property Right. So the answer here is option 2, RT graph.